Hello, everyone. It's a pleasure to talk to you today about a very exciting combination of two topics, artificial intelligence and precision oncology. So I'm Istvan Petak, your presenter of this topic, and I'm actually trained as a physician. So I graduated 28 years ago in 95, and after that I started working at the pediatric oncology department. And at that time, that was an era before the Human Genome Project. That was the era before any targeted therapies actually been developed. So we really used just chemotherapies. Luckily, most of children with leukemias actually were cured already at that time with combination chemotherapy, but already at that time we identified a subgroup of patients who harbored specific genetic alterations like BCR-ABL translocation. And we knew that that meant a different response. It meant a bad prognosis, but we still had to give the same chemotherapy to these children, and most of them died. And that was a really terrible experience that really made me do research. And I went to do research at the molecular pathology department. We isolated the tumor cells and we tried to figure out what is the molecular mechanism of drug sensitivity. And this was the era when we started to understand that any response to a therapy is regulated at the molecular level. And that was the golden age of apoptosis. And then I moved to St. Jude Children's Research Hospital for several years, where I worked in molecular pharmacology department. And we try to develop the concept of signal transduction therapies. And then after 2003, really the era of the precision oncology started, because we had some targeted therapies. And also we started to develop molecular diagnostic tests. And ever since, so in my entire career, my passion was always to somehow bring this dream of precision oncology come true. What I mean by precision oncology is to treat patients individually with targeted therapy based on the individual molecular profile of their tumors. So I teach at two universities, and I'm also founder and CEO of two biotech companies. One is developing molecular diagnostic tests, and other AI-based solutions to interpret these results. But today I will talk more about the background of this whole concept of precision oncology, and how can AI help us to bring this promise to really to cancer patients. So when we talk about precision oncology, we dream about something like this. And I apologize for the graphic picture of this tumor, but I think it's very important early on to face the enemy. So we should know what we are talking about here. And this is a child, an 8-year-old girl who was diagnosed with a very rare type of breast cancer. That was a case published by a group of scientists and oncologists at the Sloan Kettering Cancer Center several years ago. I just love this case to use it to demonstrate the power of precision oncology. So in this case, the tumor was analyzed in a very full detail. Even whole genome sequencing was performed, whole exome sequencing, transcriptome, targeted panel. And as you can imagine, hundreds or thousands of genetic alterations were identified, but then researchers filter out 3 major genetic alterations. And this one is the translocation of the NTRK3 gene, neural tyrosine receptor kinase, a mutation in the third promoter, and also a loss of P16ARF. Now, the beauty here is that we know everything we should know, I think, about these genetic alterations. So this is an example how exciting it is to merge what we learned about cancer biology in the past decades we bring it to the clinical practice. And now we can ask the question, why a patient has cancer? What is the underlying cause, genetic cause? So in this case, scientists and the team thought that, okay, we have an obvious driver here, an oncogenic driver, because there is a translocation, a fusion protein created from a gene which is a receptor kinase, tyrosine receptor kinase, a growth factor receptors, designed by nature to drive proliferation, actually during embryogenesis of the neuronal cells. But it's a driver of a signal transduction pathway which promotes growth. And then, of course, we need to have a loss in the brakes, the tumor suppressor genes, and one is we deregulate the apoptotic pathway by losing ARF. That's 100% of cancer patients have a functional loss in the p53 pathway, and then in this case it was the ARF. So we have the driver, we have the tumor suppressor, the loss of RIC, and we also know that this tumor is now immortal because a third promoter has a mutation that will dysregulate the expression of the telomerase enzyme, which can make this cell immortal. So we know how this cancer was made out of these three alterations. And of course, based on this understanding, this is what the team decided to enroll this patient to a targeted therapy trial, larotrectinib, which is designed to inhibit NTRK3 to switch off the driver oncogene and stop the cancer growth and the survival signal. And what you could see is the dramatic response to the targeted therapy after 54 days. So it's just amazing. It's like antibiotics. It's really amazing response. So the big question is, if we can do this for some patients, how can we replicate this and provide this level of care for all our cancer patients? So that's the big question. So we are all excited about AI, artificial intelligence, and how to use computational tools, AI, and machine learning in various areas of oncology, like from diagnostics, image analysis, digital pathology, drug discovery, prevention. But the big question is, can we use really AI in precision oncology, which means N of 1 treatment? Because here the difficulty is, the theoretical difficulty, it's very difficult to actually train a single algorithm to actually link each individual patient to one of the hundreds of possible treatments. It's possible to train algorithms to find correlation between certain patterns and a certain therapy, but how we can fulfill the dream of individually selected therapies. So there was a great oncologist in Johns Hopkins, who was one of the founders of Johns Hopkins, William Osler, who had this fantastic saying that medicine is a science of uncertainty and art of probability. So this is what we tackle. And when we try to decide on a therapy, we use two types of way of thinking. One is based on statistics, correlation, and the other one is based on causation. So to really understand the concept of medicine based on causation, I would like to ask you to imagine yourself that you are in an emergency care unit and you are the doctor on duty, and at midnight a patient is presented to you, and the patient complains about a terrible headache. And you go closer and you see that there is a big nail in his head. So the question is, what would you do? What would be the best therapy to this patient? Of course, because the time is short, I give you quickly the answer. Of course, we treat the patient for migraine. We all agree, since migraine is the most frequent cause of headache. So therefore, statistically, the best decision is to treat patients with headache for migraine, so drugs for migraine. So of course, it just sounds funny, and we all think it's funny because even though probably very few of us actually ever met a patient with a nail in the head. So we use not a statistical thinking. We cause a different type of thinking I call causation when we look at the nail as the actually cause of the headache, and we have a hypothesis based on our model we created about the biology of the human body that predicts, the model predicts that there is a link between the nail and the pain. So that's really the concept of precision oncology, the precision medicine. We are looking for the nails. Versus if the patient has a hat on, so we don't see the nail, we have to try different therapies and look for the best choice based on statistics. That's today. That's the basis of evidence-based medicine. We create groups of patients who share the same symptoms, and then we try different therapies, and we try to figure out which therapy has the higher statistical chance to work for one type of patient. Of course, the problem is that once we increase the number of inclusion criteria, on one hand, the clinical trial data will be more and more relevant to our patient, but the sample size is getting smaller and smaller until to the end, when we realize that basically we are facing a patient which has a unique combination of characteristics. So this concept was put on test in my life, actually in 2003, when it was actually the review of the human genome. And that year was also very interesting because there was a clinical trial of emerging, very promising targeted therapy, gefitinib, which is a tyrosine kinase inhibitor of the epidermal growth factor receptor. It was very promising because we thought that lung cancer, for example, is a cancer originated from epithelial cells, epidermal cells. So inhibiting the growth factor receptor of that cell type should be extremely effective. And that's why it was extremely shocking that this drug actually failed in lung cancer patients, in unselected lung cancer patients. So we had the highest level of evidence, a phase 3 trial level evidence that gefitinib, this targeted therapy, is actually absolutely ineffective, doesn't add anything to chemotherapy. And the same time, when this was published, we found a lung cancer patient where we identified the amplification and mutation of the EGFR gene. But that time, there was no publication which linked the response of the gefitinib to the presence of the genetic alterations. So we simply thought that this mutation and amplification in the EGFR gene is the nail in this cancer. So this is why we initiated an investigator-initiated clinical trial and treated this patient based on this molecular alteration. And of course, you can imagine the response and the excitement when we saw this patient respond dramatically. And she came out from the intensive care unit, and all her brain and lung metastasis disappeared within weeks, and she lived for more than 5 years, and she died of an unrelated disease. So this is the ultimate dream of any anybody working in the field of cancer research, oncology, and especially precision oncology. So, but still, and then of course, dozens of publications came out which established the link between these activating mutations in the tyrosine kinase receptor and the response to the targeted therapy. Still, we had to wait to a randomized clinical trial, IPAS, which was conducted in Asia, where there were plenty of mutant patients, where they randomized the patients classically in 4 arms. So patients without mutations were randomized either to receive chemotherapy or targeted therapy. On the other hand, patients who harbored the mutation also were randomized chemotherapy versus targeted therapy. And it turned out that yes, patients who harbor the activating mutations, those benefit much more from the targeted therapy from chemotherapy. So basically, we had to wait like 5 years to get this data to really have gefitinib finally registered again and being available. But anyway, at that time in 2010, I was very optimistic because I thought that the concept of precision oncology, choosing a targeted therapy based on the presence of the molecular alteration, can be combined with the concept of evidence-based medicine. We simply have to do clinical trials where the inclusion criteria is the biomarker, a biomarker which selects a group of patients with the corresponding therapy, and they are co-developed and co-registered. So we will have a companion diagnostic test approved by FDA for each targeted therapy. And then early on, we published this concept, like many of us believed in Nature Review Drug Discovery. We also foresaw the need for agnostic therapies because, of course, the same genetic alteration can occur in multiple tumor types, and then the same tumor type can be due to different type of genetic alteration. So the way I usually imagine this, the same bacteria can cause inflammation in different organs, and the inflammation of an organ can be caused by different types of bacteria. So we have to know the cause to select the therapy. So and then all these dreams seemed to came true. We had our first tumor type agnostic therapies like the immunotherapy pembrolizumab, based on the idea that in microsatellite instable tumors, you have plenty of neoantigens to be targeted by immune therapy. So, and it was proved again in clinical trials, and was approved. And also we also had the alotrectinib, the anti-RK inhibitors approved for all cancer types. So this is so exciting because that meant that now a doctor can prescribe on label a drug to a patient who has a rare type of cancer which has never been treated with the drug, but still the drug is on label based on the molecular alteration. So, and the past years was just, I thought that we are very, very close to the concept of fulfilling the precision oncology for every cancer patient, when the results of the pan-cancer analysis of the whole genome consortium was published actually in Nature, and the researchers whole genome sequenced the thousands of tumor samples and identified at least one driver genetic alterations in 95% of cancer cases. So that's so exciting that now we can say that after 2020, we can say that we can identify the cause of cancer in 95% of cancer patients. And meanwhile, we all know that the cost of sequencing is dropping dramatically, is even cheaper and cheaper and cost effective. So we can, it's really now it's possible to get access to this information for each cancer patient in clinical practice. And the number of targeted therapies have been increasing exponentially. And now actually we have a new drug approved every month. So this is all so far very exciting. And so the troubles start when you look at the actually outcome data, and you can see that if, for example, in like basket trials and platforms like NCI Match, even in the successful arms where the targeted therapies are matched to their target, the actual absolute response rates are on average around 20%. So most patients don't respond to their corresponding the drug which corresponds to the genetic alteration present in their tumor, although 20% does, and it's still great, but that's level of high failure rate is really it's a great difficulty. And also, if you look at the website of FDA, the number of biomarkers and genetic alteration which actually are approved to be used as predictive companion diagnostics test is quite limited. I think it's around 28 when I last counted. I mean, this is much less, it's not really a lot compared to the number of drugs in clinical use or the number of cancer genes we know. So what's the difficulty? So the difficulty is simply the numbers. Now we know, according to COSMIC, that there are 733 drivers genes, and there are 6 million mutations or somatic mutations have been reported in these cancer genes. And we also know from the pan-cancer genome project that on average, each cancer harbors a combination of 4 or 5 mutations out of the 6 million mutations of the 733 genes. So if we want to really do clinical trials to figure out which is the best therapy for each possible combinations of mutations and cancer types, my calculation indicated that we would need 14.4 million years. If we use real-world evidence, so 100% of the patients that participate in a data generation, it's only 100,000 years. So we need something else. So that's why when we do a molecular profiling, an NGS test at a panel, we try to interpret. There are only a couple of dozen actually genes which actually are approved companion diagnostic tests by FDA, so it's easy to interpret them clinically. What is the corresponding therapy that the oncologist can actually prescribe based on the result? But most of the tests now, comprehensive profiling tests, contain hundreds of genes, 300, 400, 500 or more, or whole exome or whole genome sequencing. So the question is how we try to interpret these results, and now we have evidence scales. So the logic is that we try to look for in the literature any data about patients' response to therapies of patients who harbor that genetic alterations, and we also look at the level of evidence which supports this link, and this is how we prioritize treatment options linked to each of these genetic alterations. So the difficulty is that although we can look up the literature, what is the best therapy for patients who harbor the same mutation, the problem is that each patient has, as I said, had multiple genetic alterations. So in a comprehensive profiling assay, usually you see 4, 5, or even more genetic alterations, and maybe multiple of them actually linked to treatment options. So for example, this is a real case. We had a triple negative breast cancer who turned out to be also BRCA mutant, but also carrying the tyrosine kinase domain mutation in the EGFR2. So of course we could look up the literature that which therapies are effective in patients who share the same BRCA alteration or alteration BRCA gene, or patients who share exactly the same mutation. And then we know that these are PARP inhibitors, But of course, we have the other type of clinical trials where patients with the HER2 amplification, overexpression, or mutation has been enrolled. And we know from these trials that, of course, HER2 inhibitors are effective in these patients. So the question is, what about patients who are BRCA mutant and HER2 mutant? So which therapy we should choose? Do we know that if each HER2 mutation causes resistance to PARP inhibitors? What was the results of patients of this subgroup of patients within BRCA mutants, and what is the actual results of these therapies in subgroup of HER2 positive patients who are BRCA mutants? And then on top of it, these patients also had other alterations like MYC amplification, RAT2, and we have therapies developed for patients who have MYC amplification. But of course, we will have to know what is the best therapy for patients who have MYC amplification, but also BRCA mutation and HER2 mutation. So to answer this question, we will need clinical trials who enroll patients with exactly the same combination of mutations, and we have to randomize them to different arms to really see which therapy is the best. So that's the difficulty. So this is why when you submit the molecular profile to different, the same molecular profile to two different molecular tumor board, there are some publications that the concordance between the opinions of molecular tumor boards is around 50%. So there's a 50% chance that one of them will recommend PARP inhibitor, or the other with a HER2 inhibitor for example, this previous patient. So the question is how we can really use AI here, because we need to create a system which somehow mimic our logic of modeling the cause and effect relationship in a network model. And this is we published as a method, digital drug assignment. And this is a knowledge representation computational modeling system with a network model, a knowledge graph with the edges represent of algorithms which calculate the relevance of that connection in correspondence to the actual case. But the beauty of the advantage of the system is that it's multidimensional, so it doesn't link the mutations to therapy motion one by one, but they try to rank therapies based on the totality of the molecular profile. So current solutions actually interpret genes one by one, and they list treatment options for each genetic alteration based on evidence which provide information about treatment responses of drugs in patients who share the same genetic alteration one by one. And these are ranked based on the evidence level of that evidence which link between that gene and the drug. But the problem is that each patient has a unique combination of these genetic alterations, and we have to rank therapies based on this totality of information based on AI models to really come up with a really individualized treatment option. So in this case, we can have a list of drugs and ranked treatment options for this patient. And actually, we did do a clinical validation of this algorithm or model. I would not say it's a single algorithm. I would say it's a model of algorithms based on the Shiva-1 clinical trial data. That was really a historic clinical trial, the first randomized clinical trial of precision oncology. And so in these patients, patients were treated based on the presence of genetic alterations, and only patients who had an actionable genetic alteration were enrolled in this study led by Professor Tourneau. And it was really pioneering this, and that was great. And and they found like around 50% disease control rate. So some patients responded, some not, but overall, the results were not better than the other arm where patients received chemotherapy. Therefore, it was like a negative trial. It didn't show any advantage to precision oncology. That's why it really set back the cause of precision. So that's why we wanted to go back to Professor Turno and analyze the data together again. And we used the molecular profiles of these patients and predicted whether the model would predict the response to these therapies. And it turned out that there is patients who actually did respond to the corresponding targeted therapy had 3 times higher aggregated evidence level. This is how we call the predictive score calculated by DDA. And also we could identify the 30% of patients actually benefited much more from targeted therapy than 50% in the chemotherapy. And also we identified patients who clearly did not derive any benefit from precision oncology. And this is just a case report, how you should imagine this in clinical practice. This was a neuroendocrine tumor, 50 years old lady who had multiple liver mets, and she was in very bad performance status, and the oncology had to make a very fast, quick decision whether to use everolimus, sunitinib of chemotherapy as a first-line treatment. And based on the liquid biopsy assay, based on the molecular profile, we ranked treatment options, and everolimus had the highest score, and actually the oncologist used this drug, and the patient had a very quick, dramatic response. Of course, this is just one case. It just shows the clinical situation where would you use such a tool. Of course, you would say that again, that these mutations in this case are not approved predictive biomarkers of either everolimus or sunitinib, and they are not designed to make a decision which one to use. But my problem is that if you want to wait to really use this type of information, we will need 14 million years, and we don't want to wait. So we need an out-of-the-box solution, how to really start to use molecular information in a way which is standardized, reproducible, and evidence-based in precision oncology. And I think the solution is to create a new class of medical devices. So we have diagnostic tools, therapeutic tools, but now we will have computational tools which are used to individually choose the right therapy for our patients. And because this is software-based, is a standardized system and model, it can be validated by evidence-based medicine method. So we are not doing a clinical trial for each individual drug or combination of drugs. We create a new type of devices with testing clinical trials to find the best device, the best method to choose the right personalized therapy. So I think by combining now with the molecular profiling and the advancement of molecular diagnostic with the new tools created by computational science and artificial intelligence, now we can develop the real concept of precision oncology and provide evidence-based, safe, and quality controlled, but yet personalized treatment to our patients. So I'm very excited to work with the community all around, and thank you for being interested in this topic, and hope to see you around and work towards this dream together. So with that, I would like to thank your attention, and see you in the real world, hopefully soon. Goodbye.